PepVEP is a new resource from the EBI, which provides residue-specific structural and functional annotations for every possible missense variant in the human proteome in order to investigate their consequences. It collates variant and functional data from multiple EBI resources and is kept up to date with regular releases. Users can submit their variants in 3 different ways. Either they can paste their variants in VCF or HGVS format into this box. They can upload their VCF file with no upload limit, or they can access the PepVEP data programmatically via the REST API. The results page shows the mapping from genomic coordinate to the protein position. By default, PepVEP shows the position in the UniProt canonical isoform, although by expanding any row you can see the positions in all the alternative isoforms. In addition to the mapping information, PepVEP also shows the codon change, amino acid change and consequence. And finally, PepVEP provides functional and structural information in 4 categories. The first category shows the functional information for the residue, the region and for the protein. The second category shows population observations, co-located variants in the same genomic position or in the same amino acid position, and also information on diseases that have been associated with that protein or with that region. Thirdly, PepVEP provides the structural data, that is the position of the variant in all known structures for the protein. And finally, PepVEP provides information on conservation and predictions such as polyphen and SIFT. In the functional tab, PepVEP splits the data into 3 major categories: the residue, the region and the protein. In the residue section, PepVEP displays information, annotations that have been found for that specific variant, for the variant which has been inputted by the user. The region section shows information on the region in which the variant is found. And the protein section contains general information on the protein, such as catalytic activity, the subunits, domains, post-translational modifications, and the protein's family. Finally, information is provided about the gene and the transcript. In the Structure tab, we can see the position of the variant in the structure. By default, PepVEP uses the representative structure suggested by the PDBE, but other structures of the same protein can be selected and it will change the image. By clicking on the variant, we can see a zoomed version and we can see atomic positions in more detail. Results can be downloaded. All annotations for the variants inputted can be downloaded. For small queries, these are returned instantly, whereas larger queries are returned via an email service. The data can be accessed programmatically, and PepVEP has get and post services in order to retrieve information for single variants, lists of variants, whole genes, proteins or regions within them. The user can use dbSNP, HGVS or VCF. This shows the various possibilities to find information via the API. We can see that a gene can be searched, a whole gene, a whole protein, or even a PDB accession here. So we can find all variants for a particular structure. The JSON's result file contains information, annotations on the canonical isoform, but also annotations for all alternative isoforms. In addition to the annotations, all primary sources and versions from the data are also displayed. In summary, we hope that PepVEP will help our users to interpret the variant effects using structure and function with the latest available data. We will have quarter releases of PepVEP to include data updates, but also feature improvements. We're currently undergoing testing, and PepVEP will be available in August of 2021.